Hi everyone, my name is Tracy Steinrücken. I'm the president of the Australasian Mycological Society and a postdoctoral fellow at CSIRO in Brisbane. Before we begin, I'd like to acknowledge the traditional owners of the lands around Australia, New Zealand and around the world on which we're all meeting today. I'm speaking to you from south of Maiwar, the Brisbane River in Queensland, on the traditional homelands of the Yagara and Yugambeh people. I pay my respects to the elders past, present and emerging. It's great to see lots of people signing in today from countries outside of Australia and New Zealand, so welcome to our seminar. Today's talk is by Professor Levi Kiss. Levi is a professor of plant pathology and the director of the Centre for Crop Health at the University of Southern Queensland, Australia. Before moving to Australia in 2017, he was the director of the Plant Protection Institute of the Hungarian Academy of Sciences in Budapest from 2012 to 2016. And he worked in that institute altogether for 25 years. During that period, he also worked for some time abroad in France, the UK and the USA. And this was with the support of Fulbright, the European Union and OECD fellowships. Levi's research has been focused on obligate biotrophic plant pathogens, especially powdery mildews and rusts, mycoparasitism, and biocontrol of crop diseases and weeds. I will hand over to Levi shortly. Just wanted to also let you know that the AGM will be running straight after Levi's talk. It starts at 1 p.m. Sydney time, so members are welcome to attend. I'll put the link for that in the chat as it is a different Zoom link. So if you'd like to ask a question or post a comment for Levi during his talk, you can do so by clicking on the Q&A button at the bottom of your screen at any time. And he will answer the questions at the end of your presentation. Alternatively, you can put your hand up and I'll flick to you and your microphone and camera. So after the discussion, take a moment to answer the short survey and that'll hopefully help us to improve the delivery of the seminar series for 2022. Thank you to Levi for joining us today. Please take it away.
Thank you, Tracy, for this nice introduction, which may explain my strong accent. So you may understand why I have this strong accent here in Australia. And thank you very much for your interest in this presentation, which is about interfungal parasitic relationships or mycoparasitism, which can be defined as a parasitic interaction between two fungal species or sometimes even between two strains belonging to the same fungal species. And just like so many other things in the 19th century, the study of mycoparasitism started with Heinrich Anton de Bary, an amazing scientist, a medical doctor and a botanist and a microscopist in the 19th century, who is considered as the founding father of plant pathology and modern mycology. As an introduction, just one drawing from de Bary's work. This is an interaction between two members of the zygomycota, and after carefully examining this interaction under a light microscope, de Bary clearly understood and documented here how one of these two species actually produces feeding-like structures, which can be called haustoria, inside the hyphae of the other species, which is the mycohost. And this is how one species, the mycoparasite, is able to thrive and live and sporulate and grow and spread using nutrients from the other fungus, which is the mycohost. There are many cases of mycoparasitism known in terrestrial ecosystems. A common one is mycoparasitism of rust fungi. Here you can see a rust pustule, uridinium, surrounded by a whitish material in which there are pycnidia of the mycoparasite, which is known as Eudalucacarisis. Although this interaction is quite common all over the world where rust fungi occur, the exact interaction between the hyphae of the mycoparasite and the rust colony is not really known. This case is much better known. This is the phenomenon of coiling when the mycoparasite, in this particular case Arthrobotrys oligospora coils around the hyphae of its mycohost and then extracts nutrients and parasitizes the mycohost colony. Now, Arthrobotrys is mainly known as the nematode-trapping fungus. However, as you can see here, it also acts as a mycoparasite. A recent review from 2019 of interfungal parasitic relationships produced this figure that actually summarizes the different types of mycoparasitic interactions. On the left, you may see the fusion type of mycoparasitism, when the mycoparasite hyphae simply fuse with the mycohost and nutrient uptake is happening through that zone. Then there are the haustorial mycoparasites. This is the case discovered by De Bory in the 19th century. And I really don't want to compare the two figures, because I guess De Bory's from the 19th century is a little bit nicer and more artistic and much more carefully executed than the figure from this review. So some mycoparasitic interactions are biotrophic, some others are necrotrophic, and some, as you will see in a few minutes, can switch from biotrophic to necrotrophic phase. A third case of interaction is the intracellular mycoparasitism, when the mycelium of the mycoparasite is mainly inside the hyphae and the whole mycelium of the mycohost. And then there are interactions called non-contact necrotrophic mycoparasitism, which to me doesn't classify as a mycoparasitism because they don't really come into contact, and the well-known coiling, which is very well known in Trichoderma species, for example. Trichoderma species and strains act as mycoparasites and have a number of other roles in the ecosystem. So this is a very schematic review of all the types of mycoparasitism. We will talk today about just one of these many interactions. And this is the interaction between plants parasitized by powdery mildews, PMs, obligate biotrophic plant pathogens of many dicots and monocots, and pycnidial mycoparasites of powdery mildews that belong to the genus Amplomyces. And I consider this interaction as one of the most evident cases of mycoparasitism in the field. Why is that? First of all, because it happens on the aerial plant surfaces. The symptoms caused by powdery mildew infections are so striking, absolutely clear, that whitish cover on the aerial plant surfaces. And sometimes it is visible even to the naked eye that there are brownish spots in the white powdery mildew colonies. And a simple microscopy will reveal that these brownish spots are actually masses of pycnidia of the mycoparasites belonging to the genus Amplomyces. This is actually a fine-tuned tritrophic interaction between plants, specific parasites or pathogens, these are the powdery mildews, and their specialized mycoparasites, which can also be called hyperparasites because they are parasites of parasites. If we won't talk about the interaction between the host plant and powdery mildews and host plants and the mycoparasites, although these are really interesting interactions. Today we will only focus on the mycoparasitic interaction within this tritrophic relationship. And there is a little figure of the life cycle of the mycoparasite in the asexual stage of powdery mildew. Their fruiting bodies, pycnidia, are produced usually in the conidiophores of the powdery mildews. And these intracellularly, these are filled with small spores of the mycoparasite, which are released on the host plant surfaces. They are splash dispersed in the phallosphere, germinate, and if there are other powdery mildew colonies, not necessarily belonging to the same powdery mildew species as the original mycohost, then they quickly penetrate powdery mildew hyphae, and then their development, their hyphal extension from cell to cell within the powdery mildew hyphae, and then their sporulation, the production of these pycnidia, happens intracellularly. Some of the hyphae of the mycoparasite actually reach the already formed conidia of the powdery mildew colony, and these can be taken by wind to long distances. So I call these mycoparasitic hyphae as hitchhiking hyphae, because these are basically transported by powdery mildew colonies to long distances. Of course, this interaction was discovered and studied by de Bory, and these are just one of their, or some of his beautiful drawings. He clearly recognized that this interaction means one fungus is inside another fungus and grows inside the other fungus. Sometimes the mycoparasite can go out of the parasitized powdery mildew hyphae, but usually they don't do that, and they just produce their intracellular pycnidia inside the conidiophores of the powdery mildews. De Bory, using simple light microscopy, has also documented how the mycoparasitic spores, those very small tiny conidia, actually germinate and penetrate powdery mildew hyphae on the right side of the screen, and also penetrate germinated powdery mildew conidia, and then the little powdery mildew colony that will develop from these already germinated conidia will be parasitized basically from day one by Ampelomyces. Under the light microscope, this is what we can see. On the left-hand side, you may see the brownish pycnidia inside the powdery mildew colony. This is what we can see under the dissecting microscope. And then with the light microscope, we see pycnidia in the conidiophores. And you may also see their conidia of powdery mildews, which actually escaped the mycoparasitic attack. They are not parasitized. So this is a general observation, and it has already been made by de Bory. Basically, these mycoparasites reduce the asexual sporulation of the parasitized powdery mildew colonies, but don't stop completely their sporulation. Moreover, their hyphae, as I just mentioned, can be taken by wind, and then they can grow out of the parasitized powdery mildew conidia, just like up there on that photo. Or if the parasitized powdery mildew conidium germinates and establishes a little new colony on another host plant surface, Ampelomyces mycoparasites will be there from day one and will slowly follow the development of the mycohost colonies. Now, the sexual morphs called Chasmotacea of powdery mildews are also parasitized by Ampelomyces, and these are completely destroyed. So no ascospores will be produced in the parasitized Chasmotacea of powdery mildews. As you see on that photo in the middle, basically young Chasmotacea, the teleomorph or the sexual morph of powdery mildews, becomes basically one single pycnidium of Ampelomyces and will be full of the spores, conidia of the mycoparasite. This figure just summarizes the life cycle of Ampelomyces, excuse me, in the asexual and the sexual stages of the powdery mildews. Basically, everything that I have already mentioned, it's there on this figure. Probably one of the most important questions that people ask when simply observing this interaction in the field was whether these mycoparasites have any impact on parasitized powdery mildew populations, or they simply slowly follow their development and don't really have anything to do with natural control of powdery mildews. To answer this question, there was a very nice opportunity in Finland where there is a unique study, long-term, probably has been running for 20 years in Finland, an extensive study of a wild plant pathogen interaction on a number of islands between Finland and Sweden, the Åland archipelago, where there are around 4,000 sites well identified with GPS coordinates that are visited every year, probably twice, by a number of students and researchers, as I said, for probably at least 20 years. And all the observations of the interaction between this wild plant, Planteo lanceolata, a common weed also here in Australia, it was introduced to Australia, and its powdery mildew. So these observations have been recorded for such a long time and were analyzed, and amazing results were obtained. Amplomyces mycoparasites are part of this interaction, so it's a tritrophic interaction, and the impact of Amplomyces on the local powdery mildew populations infecting this weed have also been studied, and it has become clear based on long-term observation, long-term data, that the overwintering survival of the powdery mildew populations on this weed is indeed heavily impacted by the presence of the mycoparasites. Some powdery mildew populations have simply been wiped out during winter, which means that in the autumn plants were heavily infected, and in the spring the young plants were powdery mildew free. And basically later during the season, the airborne conidia of powdery mildews may have re-established the infection. So yes, there is a well-documented impact of these mycoparasites on the mycohost populations, thanks to this work. Now, what happens with these mycoparasites in the laboratory? There are large culture collections of Amplomyces mycoparasites isolated from all over the world from many different powdery mildew species. There are some strains in international collections such as CBS, ATCC, and we have a number of strains isolated here in Australia from different powdery mildew species. Most of them are deposited at the Queensland Plant Pathology Herbarium. Amplomyces strains don't grow very well on artificial media, but they can be cultured, they can be sub-cultured. Some of them sporulate, and then it is possible to produce spore suspensions, conidial suspensions, which can be sprayed on powdery mildew infected plants, and mycoparasitic interaction will happen. What is important is that all these Amplomyces strains isolated from diverse powdery mildew species all over the world are phylogenetically diverse. However, the taxonomy of the genus hasn't been resolved yet. We don't have a good species concept yet. Hopefully we will have that pretty soon. But it is really important that we have a number of loci, of course, including ITS, but also a number of other loci that are used in phylogenetic analysis of the genus Amplomyces, and they all show that they are diverse. They belong to a number of molecular operational taxonomic units. And there are also ways of doing mycoparasitic tests in the laboratory using detached leaves or other means to study mycoparasitism. Now, one question is always whether the strains isolated from different mycohost species are actually specialized, narrowly specialized on the powdery mildew species where they come from or not. And there are controversial results in the recent literature. Some studies based on phylogenetic analysis found that some Amplomyces strains are associated with the powdery mildew species that actually hosted them, while other similar analysis without doing mycoparasitic tests, without checking the host ranges of Amplomyces strains, reported otherwise. So we have carried out a field experiment, a series of field experiments to actually trap Amplomyces strains that naturally occur in a number of powdery mildew species using other species, other powdery mildew species that were actually displayed in the close vicinity of the plants that have been naturally infected with Amplomyces. And we did that because based on all phylogenetic analysis, it turned out that Amplomyces strains from apple powdery mildew on the left of this picture are different from all the other strains isolated from many other powdery mildews later during the season. Now, apple powdery mildew starts its life cycle early in the spring, right after the emergence, the bud break and the emergence of young leaves. And usually Amplomyces overwinters within those buds or close to those buds, and infect apple powdery mildew colonies early in the season. Then later in the season, the apple powdery mildew epidemic slowly stops. Mature leaves are not as susceptible, mature apple leaves are not as susceptible to powdery mildew as the young ones. And many other powdery mildew species show up in the environment on other diverse plants. So when we simply put tobacco plants, potted tobacco plants and potted cucumber plants infected with other powdery mildew species on apple trees that have been carrying Amplomyces strains naturally, and we genotyped those. 2 weeks after the start of this experiment, we were already able to isolate Amplomyces mycoparasites from these trap plants. And we isolated them and we genotyped them using ITS sequences and also microsatellite markers. We did the same experiment in autumn, basically putting there the same 2 trap plants with 2 trap powdery mildew species in close vicinity of other plants that had Amplomyces strains that were genotyped by us. And what we found, it was exactly the same thing as in spring. Basically, Amplomyces strains that were genetically different quickly jumped on these new potential mycohost species. The intensity of the mycoparasitism was pretty much the same. And then we were able to re-isolate these strains and genotype them and concluded that there is no indication of a narrow mycohost specialization of Amplomyces strains that infect, for example, apple powdery mildew or those that infect another plant species. And we explained the genetic differentiation of these mycoparasites with temporal isolation of the Amplomyces populations. Now, this idea of temporal isolation has later been tested using many strains, more than 600 strains, isolated from apple powdery mildew all across Europe and many other plants all across Europe and also Asia and also North America. And the microsatellite analysis clearly confirmed the idea of temporal isolation. We have also studied the host specificity of Amplomyces mycoparasites in another study where we basically isolated as many Amplomyces strains as possible from nothing but grape powdery mildew in Italy and Hungary. And we sequenced the ITS and the actin gene sequences, and these are the results of the phylogenetic analysis there. There were 5 clades identified in these in this particular study, and basically 4 out of 5 of these clades included strains isolated from grape powdery mildew. So again, no indication of narrow specialization of these Amplomyces mycoparasites that attack grape powdery in also in other places. We have also used trap plants, potted grape plants infected with powdery mildew and exposed to different other plants, and those again trapped genetically different mycoparasites. So we are pretty convinced that there's no narrow host specialization of certain Ampelomyces strains to their mycohosts. However, it can happen because, of course, these studies should be continued. It may happen that some strains are more associated with certain mildews, such as apple powdery mildew, due to, for example, temporal isolation. Now, it's really nice to work with this tritrophic interaction, but the question is there any practical outcome of this study? So can these mycoparasites be used as biocontrol agents or biofungicide products against economically important crop pathogenic powdery mildews, and most importantly, grape powdery mildew, because that is a problem, and there is a potential to use biofungicides instead of chemical fungicides to control, for example, grape powdery mildews in vineyards. And the number of selected strains of Ampelomyces have been commercialized. The most advanced one is known as AQ10 biofungicide product, which was registered in the European Union and the USA. To my knowledge, it has also been tested in Australia, but it hasn't, the process hasn't progressed at the end. There is also a product known QFAC that was commercialized in Korea, and there are a few other Ampelomyces strains that were developed commercially and sold as biofungicide products against the conidial stage of powdery mildew. So basically against the rapidly spreading stage of powdery mildews. And the results are sometimes contradictory, not always convincing. How good are these biofungicides based on Ampelomyces against crop pathogenic powdery mildews? So we had a project back in Hungary and Italy and Spain, funded by the European Union, where we tried another strategy. We applied Ampelomyces against the overwintering form of powdery mildews against Cosmotacea in autumn, and then we checked the start of the epidemics and other patterns in spring. And we were able to show that by destroying the overwintering inoculum of grape powdery mildew in European vineyards, the epidemic can be delayed in spring considerably, and this has an economic benefit. The method, unfortunately, cannot be really used in Australia because here the overwintering of grape powdery mildew and the whole life cycle of the pathogen is quite different. However, we developed this method, and we also selected commercially useful strains which were delivered to those biocontrol companies that were part of this European Union project. However, as I said, the efficacy of using these mycoparasites in crop protection is sometimes questionable. So I'm just wondering if there is a future for Ampelomyces as a biocontrol agent of powdery mildews, because these are amazing mycoparasites. They know something that no other microbe in the phylosphere knows, and this is how to attack, how to penetrate, and how to eat up internally powdery mildew mycelia. So probably genetics and genomics should be applied, and we have already started this journey to look for other opportunities of using these mycoparasites as biocontrol agents in crop protection. First of all, we developed a molecular genetic toolbox for Ampelomyces based on agrobacterium-mediated transformation. And the first step was GFP transformation, a transformation with the green fluorescent protein of Ampelomyces strains. The transformation worked nicely, and the visualization of the transformants of the GFP transformed Ampelomyces was nicely there under the fluorescent microscope. However, I'm not sure if we actually discovered anything more compared to what Du Bois has already seen in the 19th century about the interaction between powdery mildews and Amplomyces. It's just a GFP transformant. However, these GFP transformants have also been useful in studying whether there is a niche other than inside powdery mildew colonies for Amplomyces, because that has always been the question. Are these mycoparasites restricted to the mycoparasitic way of life inside powdery mildew colonies, or they are actually also saprobes, or maybe also plant pathogens, or maybe who knows what in the ecosystem. So we use the GFP transformant Amplomyces to check their saprobic abilities in decomposing leaves in the soil and in other powdery mildew-free environments. As mentioned earlier, they can be cultured on agar media, so they have some saprobic abilities. However, we were not able to show that these GFP transformants are able to sporulate or to spread in any environment that is powdery mildew free. And when you search the NCBI gene bank, there won't be any indication of Amplomyces, for example, ITS sequence detected in any environmental samples randomly taken from different places that are powdery mildew free. So we don't have such evidence. However, the same GFP Amplomyces transformants were able, the same transformant actually was able to happily and quickly parasitize 5 different powdery mildew species belonging to different powdery mildew genera that we included in this analysis. So it looks like the saprobic abilities of these mycoparasites are really limited. So we have the agrobacterium-mediated transformation as a toolbox. And thanks to Alex Idnulm at University of Melbourne, we now know that CRISPR gene editing also works in Amplomyces. So there is a way to mutate genes also with CRISPR in Amplomyces strains. So we have these methods at the moment. And we also have genomes, high quality genomes of Amplomyces strains. One has just been published. Another one is also available in GeneBank, but it hasn't been analyzed yet. And we also have maybe 3 or 4 more genomes that haven't been published yet. So we have genetic and genomic tools to continue this journey with the study of these particular mycoparasites. And we have just started an ARC discovery project. The title is "The Enemy's Enemy is My Friend," because basically the enemy is the enemies are the crop pathogenic powdery mildew fungi, and their enemies are, of course, Amplomyces mycoparasites, and they should be our friends in case we would like to apply them in crop protection. So we started this ARC project in collaboration with University of Melbourne, Alex Idnulm's lab, and University of Zurich in Switzerland, Beat Keller's lab, to answer basically 2 important questions. One is, what makes Amplomyces uniquely capable of basically entering powdery mildew colonies silently without any obvious defense mechanism from the attacked powdery mildew colony, and then eating their cytoplasm up internally while growing from cell to cell in powdery mildew colonies. So what is the secret of Amplomyces? How can they do this? There are obviously so many other microbes around powdery mildew colonies, of course, bacteria, other fungi, protists, and so on, that would all probably enjoy powdery mildew colonies as a food source. However, none of these other microbes are actually able to exploit powdery mildew colonies. There are other microbes that may produce antibiotics like metabolites to suppress powdery mildew colonies. There are a few other antagonists known, but it's only Ampelomyces that can do such an amazing job and use powdery mildew colonies as a food source. Of course, we have speculated whether there is RNA interference or there are other mechanisms used by Ampelomyces to eat up powdery mildew colonies. We don't know. We are well aware of the structural interaction, but the mechanisms behind are not understood at all. And let's say we will learn something from Ampelomyces. We will find out some of its secrets. Can we apply these secrets to basically to use Ampelomyces derived metabolites and Ampelomyces derived products? For example, I'm just speculating here, small RNAs to be used in the control of crop pathogenic powdery mildews. So that would be, of course, the ultimate goal of this project. And this may mean that, yes, there is a future for Ampelomyces mycoparasites as a biocontrol agent of economically important crop pathogenic powdery mildews. So this project has just started. One interesting outcome was the analysis of the first genome. And surprisingly, this genome showed patterns, so-called bipartite structure, that pointed to the genomes of well-known plant pathogens, including Leptosphaeria, including Parastagonospora, Pyrenophora. These important crop pathogens are phylogenetically closely related to Ampelomyces mycoparasites. This is another secret that has to be revealed. How come that a fungus that has relatives that are well-known crop pathogens has just changed and became a strict mycoparasite of powdery mildews and not showing any patterns of plant pathogenicity anymore? So the project has already started and has already produced this interesting first result, but of course there are almost 3 more years to complete this study. And we have a 2-year postdoc position and also a full-time PhD position within this ARC discovery project. So in case you are interested or you know someone who is interested, just please contact me. The postdoc position is advertised at our university's website as we speak, and it will be open for probably for less than 2 weeks from now. This is where I would like to conclude, and I would like to thank so many people who contributed to all these studies on Ampelomyces mycoparasites. Probably I won't list them because we are short of time. Is this correct, Tracy? I'm just should I stop here and?
No, still got a bit of time if you want to mention anyone in particular. That's fine. Okay,
so I'm really grateful to my colleagues here at the University of Southern Queensland. Most importantly, Nilufar Vagifi, Gavin Ash, and Lauren Hutt. Also to my colleagues back in Hungary, Gabor Kovács at Eötvös Loránd University, and also Mark Németh and Alexandra Pintje at the Hungarian Academy of Sciences. My collaborators in Italy, Tito Caffi, Sara Legler, and Vittorio Rossi. UPay 10 at Biosecurity Queensland. Kalman Zoltán Váci at Eszterházy University in Hungary, Markus Gorfer at Australian Institute of Technology, Tatiana Giraud at University of Paris, Annalisa Lane at University of Helsinki, Beat Keller at University of Zurich, and Hyandong Shin at Korea University, and a number of other colleagues who all supported these projects. As you may have realized, I have talked here about many projects done during the past probably 15 years, and these have been supported most recently by our university and ARC, and earlier by the Hungarian Scientific Research Fund and also European Union project. So thank you for your attention.
Thank you so much, Levi. That was fascinating. I come from a biocontrol background, so it's really interesting to see your side of the research at the moment. We do have one question, but I'm going to kick off with one of mine first. You said that the pathogen life cycle of powdery mildew is different in the EU and USA compared to Australia. But I was just wondering about other climatic zones. I mean, obviously here in Queensland, it's one thing, but is there any similarities between those climate zones in Europe and here where this could be used?
Yes, I'm thinking of Tasmania, for example, because basically these studies about temporal isolation were done in the temperate zone, where we have the 4 seasons and harsh winters, most importantly. During harsh winters, powdery mildew mycelia usually don't survive. The only survivor structures are the teleomorph or the sexual morphs with ascospores. Now, maybe in Tasmania is the same or similar situation. However, if grey powdery mildew also overwinters as a mycelium in the buds, then probably there's no reason for temporal isolation, because other powdery mildews will also be there in the environment.
Right, awesome. So Peter Johnson's asking, has anyone looked for Amphilomyces-like fungi on other Letiomycetes phylogenetically close to powdery mildew?
Yes, and so there were extensive studies like that. That would be another lecture, I guess, because there's also a very interesting and I would say sad story of misidentification of Amphilomyces strains. So many strains were thought and were actually deposited and published as Amphilomyces, but those are basically other pycnidial fungi within the Leotiomycetes. Yes, so basically the answer is no. But there are contrasting results in the literature due to those misidentification problems that have also been published.
Excellent. Joanna Wong says, thank you for the great talk. Is Amphilomyces related to other non-hyperparasitic fungi phylogenetically?
Yes, as mentioned, they are closely related to Leptosphaeria, to Parastagonophora and Pionophora. These are all crop pathogens. To other pycnidial fungi, also to other mycoparasites, such as Paraconiotherium and also Eudarluca, the rust mycoparasite. So phylogenetically, Amphilomyces is quite closely related to both other mycoparasites and crop pathogenic fungi. Interesting.
Yeah. And another question from me, are there any other biocontrol solutions based on mycoparasites being developed in Australia that you know of, other than Amphilomyces?
Against crop pathogens, other powdery mildews, I mean. You mean?
Yes.
Yes, of course, yes. There are a number of biocontrol agents commercially available. Not just fungi, but also bacterial strains such as Bacillus have been developed and are commercially available as biocontrol agents.
Okay. Are they regulated in Australia through the APVMA, or is it that? No, not yet.
Yes, no. I hope they are. I hope they are. Yes.
Trying to develop a mycoherbicide. I know they are regulated, but I wasn't sure about others. Okay. D. Carter says, thanks for the very interesting talk. Is there a reason that you favor RNAi over other secreted factors in the interaction with the host?
we thought of RNAi just because it hasn't been explored at all in any mycoparasitic interactions so far that I am aware of. And of course, it is important. RNAi is involved in the host plant powdery mildew interaction. So host plants produce small RNAs to protect themselves from powdery mildews. So we thought that maybe this could be a kind of indication that the mycoparasite can also use RNA, small RNAs, to somehow to silence the defense mechanisms of powdery mildew mycelia. Now, nothing is known about the defense mechanisms of powdery mildew colony. We don't know that. So that's also included in this project. Mm-hmm. Talk more about that.
Jeff Powell also said, thanks for the excellent talk. Are there any plant factors that affect Amphomyces fitness other than those that would increase susceptibility to powdery mildew?
Well, when it was about developing new strains to be applied as biocontrol agents, then we compared the fitness of a number of strains in terms of how quickly they spread inside the powdery mildew mycelium, how well they sporulate, and basically we concluded, and that was even the title of one of our papers, that what was important from a biocontrol perspective is how well a certain strain sporulates in culture. Because once they sporulated in culture, there were no obvious fitness differences when different powdery mildew species were challenged with the same Amphomyces strain. So yes, it's a very good question. Probably much more should be done in this space.
Excellent. And I'm just seeing Alex Idnam is answering Joanna Wong as well, that Amphomyces is within the Leptosphaeriaceae family, so related to the plant pathogens in the genera Leptosphaeriaphoma planodomus, or whatever the favorite name is at the moment. Okay, well, I'm going to end it there so that we can give people a break for those who are going to be joining us for the AGM on the hour. Thank you so much, Levi, for joining us today and for the interesting talk. We look forward to seeing how those postdocs and PhD projects turn out, and hope to hear about those in a seminar next year sometime. But thanks everyone for joining us, and look out for the next talk. We're going to be having a special event one on the first Wednesday of December, featuring research grant awardees. So we'll see you then.
Thank you very much. Bye. Bye everyone.